Hello everyone. In this video, we will discuss about the enzyme-linked immunosorbent assay that is also called as a ELISA or ERA. Is a principle to RIA but depends on an enzyme rather than a radioactive label. An enzyme conjugate with an antibody react with colorless substrate to generate a color reaction product. Such a substrate is called as a chromogenic substrate. A number of enzymes have been employed for ELISA, which including alkaline phosphatase, then horseradish peroxidase, beta-galactosidase, etc. This assay approach the sensitivity of RIA assay and have the advantage of being safer and less costly. Next one is the types of the ELISA, that is also called as a variation of the ELISA. So there are numerous variants of ELISA which include indirect ELISA, then sandwich and competitive ELISA. These number of variations of ELISA have been developed, allowing qualitative detection or quantitative measurement of either antigen or antibody. Each type of ELISA can be used qualitatively to detect the presence of antibody or antigen. Alternatively, a standard curve based on known concentration of antibody or antigen is prepared from the given unknown solution for sample can be used. Antibody can be detected or quantitatively determined with an indirect ELISA. Serum or some other sample containing primary antibody is added to antigen-coated microtiter well and allowed to react with antigen attached to the well. After any free antibody burn is washed away, the presence of antibody bound to the antigen is detected by adding an enzyme conjugated secondary antibodies. So, these secondary antibodies bind to the primary antibodies, and if there is any free antibody too, that is the secondary antibody, then it's washed away and a substrate for the enzyme is added. The amount of the color substrate, so you can see here, okay, the amount of the color reaction produced that form are measured by the specialized spectrophotometric plate reader, which can measure the absorbance of all the wells of 96 well plate in seconds. This indirect ELISA is method of choice to detect the presence of serum antibody against the immunodeficiency disease or the virus which is responsible for that is the HIV. Next one is the sandwich ELISA. So antigen can be detected or measured by sandwich ELISA. In this technique, the antibody rather than the antigen. Okay, so what is the difference between indirect ELISA and sandwich ELISA? In indirect ELISA, antigen coated well, the plate which is used is coated with the antigen, and here in case of sandwich ELISA, plate which is used for the detection is coated with the antibody. Okay, so these antibodies are coated on the well. A sample containing antigen is added and allowed to react with equine antigen. Okay, after the well is washed, okay, a second enzyme link antibody specific for a different epitope on the antigen is added and allowed to react with the bound antigen. After any free second antibody is removed by washing. Okay, after that color reaction is produced, which is read by the same as in the indirect ELISA by the spectrophotometric plate method. Okay, this is the second method, that is the sandwich ELISA. Next one is the competitive ELISA. Okay, so in case of competitive ELISA, is used. So, another variation for measuring the is amount of antigen is the competitive ELISA. In this technique, antibody is first incubated in a solution with a sample containing the antigen. Okay, so after that, the antigen antibody mixture is then added to antigen coated microtiter well. The more antigen present in the sample, the less free antibody will be available to bind the antigen coated well. So this is the main principle of the competitive ELISA. Addition of enzyme conjugated secondary antibody specific for the isotype of the primary antibody can be used to determine the amount of primary antibody bound to the well as in an indirect ELISA. In the competitive assay, however, the higher the concentration of antigen in the original sample, then there is the lower the absorbance. This is the basic difference between indirect sandwich and the competitive ELISA. There are also some additional techniques is developed. That is the modification of the ELISA, which include the chemilucences. This provide a convenient and highly sensitive alternative to absorbance measurement in ELISA assay. In version of the ELISA using the chemiluminescence as a luxogenic, that is a light genetic substance take the place of the chromogenic substrate. For example, oxidation of compound luminol by addition of hydrogen peroxide and the enzyme Horsch redox peroxidase produce the light, and this light is measured. Okay, the advantage of this technique over the ELISA is that it enhances the sensitivity. In general, the detection limit can be increased at least tenfold by switching from a chromogenic to luxogenic substrate. There is a one more variation in the ELISA, which include ELISpot assay. Okay, this ELISpot assay allows the quantitative determination of the number of cells in a population that are producing the antibodies specific for the given antigen or an antigen for which one has a specific antibody. In this approach, the plates are coated with the antigen recognized by the antibody of interest or with the antibody. Okay, this is the one more variation of the ELISA, that is called as a ELISpot assay. Thank you.